Welcome to the Notre Dame Linked Experimental Ecosystem Facility in St. Patrick's County Park, otherwise known as ND LEAF. My name is Elise Snyder, and I'm a grad student in the Tank and Lamberti labs at the University of Notre Dame. In this video, I will tell you all about our environmental DNA experiments in the ND LEAF streams. But before we get our feet wet, let's take the time to answer one question. What is environmental DNA? Environmental DNA, or as it is more commonly called, eDNA, is a relatively new technique developed in 2008 for detecting and monitoring aquatic organisms. An organism living in the water sloughs DNA into the environment in the form of scales, mucus, skin cells, excrement, and even reproductive cells. Basically, everything you don't want to think about when you accidentally swallow some water while you're swimming. This DNA can be extracted from water samples and analyzed to determine what is living in a body of water. Although it is a relatively new discipline, eDNA is already proving to be an excellent technique for monitoring freshwater systems, and it is especially useful for keeping tabs on endangered and invasive species. The problem is that eDNA becomes a more complicated method to use when the DNA is being transported downstream by flow, like in rivers and streams. We still don't know enough about how eDNA is transported and degraded in these systems, and that's where the ND LEAF streams come in. For our eDNA experiments, we wanted to investigate how biofilms impact the transport and degradation of eDNA. A biofilm is a slimy film of bacteria that grows on surfaces in the stream. If you've ever gone wading in a rocky stream in the summertime, you'll know all about how slippery biofilm can be. From our past research, we know that eDNA is removed from the water faster when more biofilm is present, but not all biofilms are the same. Some biofilms are made up primarily of autotrophic organisms that derive their energy from the sun, while others are mostly heterotrophic and must consume other organisms to stay alive. The different types of biofilms thrive under different substrate and light conditions in streams, which we predict will impact the transportation and degradation of eDNA. We started off our experiments by covering the leaf streams with shade cloths to prevent the growth of autotrophic biofilms. If you need the sunlight to produce energy, then you can't grow when there's no sun. Then we uncovered the streams and allowed autotrophic biofilms to develop for several weeks. Then we covered the streams back up and allowed the autotrophic biofilms to die off. During each phase of our experiment, we also collected substrate samples so that we can measure the amount of biofilm in the streams. During each of our shading phases, we added eDNA into the streams and measured how much was left as it moved farther and farther downstream. Rather than adding fish directly to the leaf streams, we collected eDNA in the form of fishy water from the Potawatomi Zoo. To do this, we simply scooped buckets of water from their moat where they house a large population of common carp. Then we mixed it thoroughly while using a pump to slowly and steadily drip the water into the stream. Since the leaf streams are fed with fishless groundwater, we know that our stream started out free of carp eDNA. After we sampled throughout the streams, we took the water samples back to the lab and filtered them so that we could extract and quantify the eDNA in each sample. These experiments at LEAF will help us reach our long-term goal of making eDNA a more useful tool in streams and rivers. I hope you've enjoyed learning about eDNA today. Thanks for watching!